Good morning, children. Welcome to our next class. In today's class, we are going to study about advancement in the modern biotechnology. Now, already we have studied the difference between the modern biotechnology and the conventional biotechnology. In that, we have studied the modern biotechnology based on the ability to change the genetic material of an organism. So, or the modern biotechnology deals with the gene manipulation. So today we are going to study about the gene manipulation method, and one method for that is the genetic engineering. So already we have studied modern biotechnology. It embraces the genetic manipulation. So here either we are manipulating the genome or we are fusion of the two desired protoplasm we are doing. As a result also the genomes they are being altered, or already existing conventional techniques they are being modified by the application of new techniques over it. So today we are going to study about the genetic manipulation, or this genetic manipulation we can also be called as the genetic engineering, or it is also called as recombinant DNA technology or gene cloning. So here in this what is being done, we are modifying the uh, one organism's genetic material by transferring the desired gene. Okay, by the transfer of a desired gene from one organism into another organism, we are modifying that organism. So the definition for the conventional recombination we have already studied. The conventional recombination techniques we have studied. It is done by involves the exchange of the genes or recombination of the genes between the homologous chromosomes in the meiosis cell division. But the modern technology, it is what we are doing, we are combining, we are obtaining the recombinant DNA, and it is done how? By the transfer of the desired gene, the DNA which is coding for a specific gene, that is being isolated from one organism and it is transferred to another organism. For this we require some agents. So that agents, it may be some vectors that is the carriers of this desired gene, or we use some instruments for the transfer of this desired gene into an organism. So the instruments what we are using for the transfer into the desired organism is some electroporation, that is we are creating some pores in the cell for the introducing the desired gene, or by the gene gun method, that is directly the desired gene is transferred into the target site, or we are following the liposome mediated method or chemical mediated transfer, or it is done by the micro injection. Now, what are the steps involved in the recombinant DNA technology that we are going to study here? So, as we have studied, the genetic engineering is the gene manipulation. So, how this gene manipulation is done, that we are going to study. As a result of this gene manipulation only, we will get a recombinant DNA. So, first step is what? Isolation of a DNA fragment containing a gene of interest that need to be cloned. This is called as an insert. So first what we are doing, we are collecting, we are identifying, or we are separating, isolating a desired gene from an organism. So that desired gene is found in an organism, that gene has to be cloned, that gene has to be multiplied. So we are first identifying that and we are isolating that desired gene, and this is called as an insert. The desired gene what we are going to introduce in another organism, that is called as the insert. Second step is generation of the recombinant DNA molecule by insertion of the DNA fragment into a carrier molecule, that is called as vector. So as we have already studied, for the transferring this desired gene we require a carrier. So that carrier is called as the vector. Okay, so with the help of this vector, the desired gene is being introduced inside a host cell. Third step is selection of the transformed host cells that is carrying the recombinant DNA and allowing them to multiply, thereby multiplying the recombinant DNA molecule. So the already now we have what we have done, we have inserted the desired gene into an organism. So that desired gene we have introduced inside the host cell. So we are introduced inside the vector. So we are getting a recombinant. That recombinant we have introduced inside a host cell and we are allowing it to multiply. As a result, we will get cells with the recombinant DNA. Next step is what we are the entire process thus generates either a large amount of recombinant DNA or large amount of proteins expressed by the insert. So here, as a result of this introduction of the desired gene into a host cell and application of the allowing them to multiply, what we will get? We get a large number of recombinant DNAs, or we will get the particular protein for why we have done this, selected the desired gene and we have introduced inside the host cell. Next step is what wherever vectors are not involved, the desired gene is multiplied by PCR technique. The multiple copies are injected into the host cell protoplasm, or it is shot into the host cell protoplasm by shotgun method. So for PCR is what it is. The polymerase chain reaction is a common laboratory technique used to make copies of a particular region of a DNA. So what you are doing with the help of this PCR, a particular sequence of gene, so desired gene, when we are not able to introduce with the help of a vector inside a host, what you are doing with the help of this PCR, we are multiplying that sequence of DNA, and then we are directly injecting into the target site inside the host cell. So this we have so this is the steps involved in the gene manipulation. So what we have studied, first we are selecting the desired gene, then second introducing the desired gene inside a vector. As a result we are getting a recombinant. Then this recombinant is inserted into the host cell and allowed to multiply to get a numerous copies of that. Okay, as a result of this multiplication we'll get a recombinant DNA copies, or we'll get the particular proteins which is secreted by this recombinant DNA. Sometimes what will happen, this technique is not possible, the insertion of the gene with the help of a vector is not possible, so we follow the PCR method. By PCR method is what a particular DNA sequence, they are being allowed to multiply, the copies are being produced. As a result, that particular copy is injected directly into the host with the help of some instrument. So here you can see the diagrammatic representation of these steps, the steps involved in recombinant DNA technology. So first what we have studied, we are taking the DNA of chromosome. So first what we are doing, the select gene of interest, we are selecting the cell containing gene of interest. That gene we are isolating, we are taking out from that cell. Okay. Next, what we are doing, we are treating it with an enzyme. We are taking a segment of that and introducing inside a vector. This vector is mostly we select a plasmid, extra chromosomal circular DNA found in the bacteria. So the plasmid acts as a vector, a carrier of this desired gene. So this desired gene is inserted. This desired gene is treated with the help of some enzymes. As a result, we get in small fragments. Similarly, the vector is also treated with this enzyme. So what is happening, this vector is cut down, and in that particular region we are inserting the desired gene. Now this vector is having the recombinant DNA, and this is reinserted inside the bacterial cell. So the bacterial cell contain the recombinant DNA. So that is bacteria is called as the recombinant bacteria. Now this is allowed to the cells clone with gene of interest and the identification of the desired clone, and it is allowed to multiply. Next, various application, these clones, for what purpose we are using, whether it is used for the toxic cleaning purpose, or it is used in the research work, or it is used for the pest resistance that we are using it. So these are the steps involved in the genetic engineering that we are going to study now in detail. So here the tools for the genetic engineering. Now we know from the foregoing discussion that in order to generate recombinant DNA molecule, certain basic tools are necessary for this process. The basic tools are enzymes. So here, as we have studied, for the genetic engineering or gene manipulation, we require some basic tools. Okay, so what are the tools required? So the three basic tools required are some enzymes. Next, we require a suitable vector, and next we require a suitable host organism where we can multiply it. The most important enzymes required for genetic engineering are the restriction enzymes, DNA ligase and alkaline phosphatase. So as we have studied, for the genetic engineering to be done, we require the enzymes. So basically we require three major types of enzymes. One is the restriction enzyme, the next one is the DNA ligase, and the third type of enzyme is the alkaline phosphatase. So let's study about each enzyme in detail now. So first one, the restriction enzyme. The two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli was isolated in the year 1963. So this restriction enzyme was isolated in the year 1963. One was the enzyme which added methyl group to DNA, while the other it cut the DNA. This later was called as the restriction endonuclease. So the function of this restriction endonuclease is what? It cuts the DNA. The restriction enzyme or restriction endonuclease is an enzyme that cleaves DNA into fragments near specific recognition sites within the molecule, that is known as the restriction site. Now what is the role of this restriction enzyme? As we are studying, there are certain enzymes. In that first we are studying about the restriction enzyme. So the restriction enzyme, they are the enzymes which cuts the DNA at specific site. That specific site where the DNA is cut with the help of a restriction enzyme, that is called as the recognition site, or it is also called as restriction site. So in this particular site only, with the help of this restriction enzyme, we can cut the DNA. Based on the mode of action, restriction enzymes are classified into two types. They are exonuclease and endonuclease. So as we have studied, the function of restriction enzyme is what? To cut the DNA. So this, how it is, where it is cutting the DNA, based on that it is classified into two types, exonuclease and endonuclease. So exonuclease are the enzymes which remove nucleotides one at a time from the end of the DNA molecule. So in this DNA sequence, it's a polypeptide chain. Now here, so from the end, from one end, one by one, the nucleotides are being removed. Then this enzyme is called as exonuclease, restriction exonuclease enzyme. So restriction exonuclease enzyme, it will remove one nucleotide at a time from the end of the DNA molecule. So example for restriction exonucleases are BAL31 and exonuclease 3. Next is the endonuclease. Endonuclease are the enzymes which breaks the internal phosphodiester bonds within a DNA molecule. So here endonuclease, what it does, it also cuts the DNA. How? By breaking the bond between the two nucleotides. Okay, between the two nucleotides in the DNA molecule. So example for endonuclease is Hint2, E-Core1, PVUL, then BAM-HI, then TAQ1. So here restriction endonuclease, restriction enzymes, they are of two types based on how they cut the DNA, exonuclease and endonuclease. So exonuclease, it cuts the DNA at end and by removing one nucleotide at a time, whereas endonuclease, it cuts the DNA in the center by breaking the bond between the two nucleotides in the DNA molecule. The restriction endonuclease, it is also called as molecular scissors. The restriction enzymes are called as the molecular scissors. These act as foundation of recombinant DNA technology. These enzymes exist in many bacteria. So here restriction endonuclease is the enzyme which is called as the molecular scissor and is essential for this recombinant DNA technology. This is obtained from many bacteria where they function as a part of their defense mechanism. Now these restriction endonuclease, they are found inside some bacteria. These restriction endonuclease in the bacteria, they meant for the self-defense. They help in protecting the bacteria from the attack of some pathogens. There are three main classes of restriction endonuclease, type 1, type 2 and type 3, which differ slightly by the mode of action. Only type 2 enzyme is preferred for use in recombinant DNA technology. So here we have studied restriction endonuclease. There are of three types. The most commonly used enzyme for the genetic engineering is type 2. And Type 2 enzyme is preferred for use in recombinant DNA technology as they recognize and cut DNA within a specific sequence typically consisting of 4 to 8 base pairs. Now this type 2 enzyme, restriction endonuclease enzyme, it can cut the DNA sequence consisting of 4 to 8 base pairs. So small sequence of DNA can also be cut with the help of the type 2 restriction endonuclease. Examples of certain enzymes are given here, so that we will be studying about the different types of type 2 restriction endonuclease we are going to study here. So first one, restriction enzyme ALU1. So this bacteria from where we are obtaining this, the microbial sources are Orthobacter luteus. So here the recognition sequence. So this enzyme, it identify the sequence from 5 to 3. You can see AGCT. So what it will do, similarly the complementary strand of 5 to 3 end is TCGA. So what it will do, it will cut the sequence between AG and CT. It will cut the sequence with the in a blunt ends. So we'll get the blunt end fragments. So symmetrical ends we will be getting here. Next type of type 2 enzyme is what? BAMH1. The source is Bacillus amyloliquefaciens. So here the sequence of nucleotides are from 5 to 3 end GATCC, and the complementary strand sequence is CCTAGG. So what it will do, this restriction enzyme BAMH1, what it will do, it will cut the DNA sequence from 5 to 3 end from G. Okay, so in the 5 to 3 end it is cutting after the G, and similarly into 3 to 5 end also it will cut the sequence after the G. So here what we are getting, a sticky end. Okay, so L-shaped ends we are getting. So this is the such type of ends we call it as a sticky ends. Next is the E. coli. E. coli enzyme, restriction enzyme obtained from the Escherichia coli. In the sequence of nucleotide is given here. Here also this enzyme, restriction enzyme, when it cuts, we get the DNA sequence in 2 fragments we are getting with sticky ends. Next one is HAE3. So Haemophilus aegyptus. So here also when this restriction enzyme, it cuts the DNA, we get a blunt ends. Next is what? The HINT3. So it is obtained from Haemophilus influenza. So here the sequence of nucleotide where it is acting is given here. So here this is the restriction sequence, and when this enzyme cuts this cuts at the recognition site, so again we will get a sticky ends here. So here we have studied about the different types of type 2 enzymes. The restriction enzyme, so HINT2, always cut the DNA molecule at a point of recognizing a specific sequence of 6 base pairs. So as here an example, that is HINT2. The DNA sequence, if its length is of 6 base pairs, this HINT2 enzyme, it can cut this base pair. The sequence is known as the recognition sequence. Today, more than 900 restriction enzymes have been isolated from over 230 strains of bacteria from different recognition with different recognition sequence. So here till now about 900 restriction enzymes have been discovered from 230 strains of bacteria, and this 900 restriction enzymes, they can cut the bacteria at different recognition sequence or different recognition sites. Restriction endonuclease, as we are saying, they are named as HINT2. So the names, how it is given? It is given by a standard procedure. The first letter of the enzyme indicates the genus name. So the H first letter, it refers to the genus name. Suppose if we take the enzyme E. coli, okay, so that E refers to the genus Escherichia. Okay, the next followed by the first 2 letters of the species, then comes the strain of the organism. So here you can see first one, E is the genus name, next CO, it is the species, R is the strain, then comes the strain of the organism, next the number I, it refers to the Roman numeral, that is Roman number 1. So here for example, if we talk about E. coli, it from the Escherichia coli E1. So here what will say E, that is the E refers to what? It is the Escherichia. Next CO refers to coli. Strain is R by 13. For that we are writing R. And from endonuclease 1 to be discovered. This one is the number of strains discovered in the laboratory. It contains two different antibiotic resistant genes and recognition site for several restriction enzymes. The sequence is referred to as a restriction site and is generally palindromic, which means the sequence on both the DNA strands at this site read the same in 5 to 3 direction and in 3 to 5 direction. So here these restriction enzymes, they cut the DNA at particular site, that is called as the restriction site or the restriction sequence. So here usually this restriction sequence, they are palindromic. So what is this palindromic? If we read this sequence of DNA from 5 to 3 end or from 3 to 5 end, they are the same. Then this sequence is called as what? The palindromic. For example, here it is given the word Malayalam. The word Malayalam is we read from right to left or from left to right, and it is the same. The sequence is called as the palindromic. So palindromic repeats what they are? A symmetrical repeated sequence in DNA strands, that is from 5 to 3n or from 3 to 5n. The same sequence is there. The sequence of the base pair in the reverse direction when compared to the first sequence. So whether it is read from 5 to 3 or from where it is read from 3 to 5, they are the sequence is, they are the reverse, and they are in the reverse direction. The exact kind of cleavage produced by a restriction enzyme is important to design the genome cloning experiment. Some cleave both the strands of DNA through a center, resulting in blunt or flush ends. As we have studied here, this restriction enzyme, they cuts the DNA at the restriction at particular site or restriction site or recognition site. So sometimes they cut the DNA in blunt ends. Okay. So or sometimes they cut it as a sticky ends. They are known as the, if they are cut blunt ends, then we call it as a symmetrical cuts. Some enzymes cut in a way producing protruding and recessed ends. They are known as the sticky or cohesive ends, and such cuts, they are called as staggered or asymmetrical cuts. As we have studied, the function of the restriction enzyme is what? To cut the DNA at a particular site. So that they cut the DNA in two ways. Either they cut the DNA in symmetrical form or it will cut the DNA in asymmetrical. So symmetrical cut, if it is, then it will have the blunt ends, or it will also called as the flush ends. For example, you can see here figure 4.5, the first row diagram. You can see the first sequence, that is in the 5 to 3n GAATTC. Okay, when it is treated with the Escherichia coli, so what will happen? The two strands, okay, as we know DNA is a two strands, the two double stranded. So here the two strands, how it is cutting? GCTTAA. Okay, and the other strand, what we will get? The other strand, how it is the complementary strand, how it is cut? AATTCG. So this end we call it as a sticky end. Next example is CCCGGG. This is from 5 to 3n. If it is treated with a restriction enzyme, so what we are getting? CCC and GGG, they are being separated by a vertical cut. Now this cut is called as the blunt ends. Okay. Two other enzymes that play an important role in the recombination DNA technology are DNA ligase and alkaline phosphatase. So we have already studied about the function or action of the restriction enzyme, how this restriction enzyme they act. So we have studied restriction enzyme, they cut the DNA at a particular site or recognition site, and we have studied there are on the basis how they cut. They are of two types, exonuclease and endonuclease. Next we have studied there are different types of restriction enzymes secreted by the different bacteria. So we have studied there are three types. Among them, type 2 is the most commonly used for the genetic engineering. The examples we have studied and how they cut the DNA sequence also we have studied here. Next, we are going to study about the next type of enzyme which has a role in the genetic engineering is DNA ligase. DNA ligase enzyme joins the sugar and phosphate molecules of double-stranded DNA with 5PO4 and 3OH in an adenosine triphosphate dependent reaction. This is isolated from T4 phage. So here the next one what we have said is the DNA ligase. Now this DNA ligase, what it does, it helps in joining the cut ends. Okay, so how this joins the cut ends? By joining the sugar and the phosphate molecules, okay, of the double stranded DNA. So here you can see the DNA ligase reaction. DNA strand with 3 end is there and another DNA strand with 5 end is there. So here DNA strand with 3 end, at the end where they have the OH, and the next they have what, the PO4 is there. So the bond is developed between this OH and PO4. Okay, so there is a ligase is done here. So what is happening in an adenosine triphosphate dependent? So for this they require a DNA, they require an ATP. An ATP is utilized here for this bonding, and as a result we get a phosphodiester bond between the 3 end and 5 end of the DNA strand. So the next enzyme what we have studied is the DNA ligase. So DNA ligase, what it does, it joins the cut ends of the DNA, and these two cut ends, they should be having the 5 end with PO4 and the 3 end with OH. So this OH and PO4, they are bonded with the help of a phosphodiester bonds with the help of utilizing one ATP. Next enzyme is what? It is the alkaline phosphatase. It is a DNA modifying enzyme and adds or removes specific phosphate group at 5 terminus of double stranded DNA or single stranded DNA or RNA. Thus it prevents self ligation. This enzyme is purified from bacteria and calf intestine. So the next enzyme is what? The alkaline phosphatase. Now this alkaline phosphatase, it is a DNA modifying enzyme, and what it does, it adds or removes some specific phosphate group from the 5 end of double stranded DNA or single stranded DNA or from the RNA, and thus what it will do, it prevent the ligation. So the self joining of is not possible here. The DNA, the particular sequence, when they come only, they can be joined. So this prevention of joining is controlled with the help of alkaline phosphatase. So this enzyme is obtained from the some bacteria, or it is obtained from some calf intestine.